Okay. Okay, so the first one is germline therapy, and the second one, guys, this is going to be somatic cell therapy. Okay, so as the name suggests, okay, germline therapy and somatic cell therapy. Okay, both of them, they are like, quite evident of what they are going to be like. So see, guys, when we talk about germline therapy, germline is nothing but it is going to be the initial cells of your body, right? So in this thing, what is going to happen in germline therapy? We modified the defective genes. Okay, so this, whatever the defective genes are, you modify the defective genes in the embryo itself. Okay, so either you're going to target the embryo, or you are going to target the sperms, or you're going to target the ovum. Okay, so that way. So basically, you are going to target the germ line, meaning your initial stages of life itself. Okay, but now on the other hand, guys, we have this somatic cell therapy. Now, somatic cell therapy, as the name says, somatic cells meaning what? Body cells. So here you are going to see that the body cells who are defective, you are going to introduce genes into the defective cells. Okay, and you're going to try to treat them. So here, what is going to happen? The body cells, they are going to be defective. And now, what are we going to do? The gene of interest, this is introduced into the defective cell. Okay, right in their location. So, guys, let's imagine. Okay, so for example, we have our pancreas. Okay, so let's say in pancreas, you are having your islets of Langerhans. Okay, but now in your islets of sorry. Okay, in your islets of Langerhans, let's say the beta cells, they are defective. Okay, so let's say your, okay, these are islets of Langerhans. Okay, and in this, the beta cells, they are defective. So now what are we going to do, guys? We are going to take the gene of interest. Okay, so let's say this is your insulin gene. This is gene of interest. You take this gene and you're introducing it right into its place to treat it. Okay, somehow or the other, you're going to put it into the somatic cells, and these somatic cells, then now, because they have incorporated the actual gene, now they will start producing the product of your interest. This is how it is going to be done. And guys, for what purposes do we use this thing? Like I said, we have the insulin thing, so we can use it for insulin. We can use it for rheumatoid arthritis. Okay, rheumatoid arthritis is RA factor. If you know, like, here you're going to have immense amounts of joint pain. Okay, so severe combined uh, immunodeficiency. Okay, so this is another disease. Then we have sickle cell anemia. Then we are going to have hemophilia. Okay, emphysema, thalassemia, etc. Okay, so these are the different types of diseases which can be treated by using this methodology. Okay, but now, guys, the next thing is, okay, I am saying that, okay, the gene of interest will be delivered into the cell, but how can we deliver it? Like, what can happen out over here? Like, what is the purpose? Like, how can we, like, target the cell itself? Okay, so let's try to see how is the gene going to be delivered into the cell. Okay, so now the first thing, guys, the first thing we are going to have is something that is called as virosomes. Now, see, guys, virosomes we had talked about over here also, somewhere out over here. Yeah. So see, somewhere here. Yes. So here, deliver bacterial or viral genes. Okay. Now this in the form of vaccination, guys. Here we can make use of this virosomes. So how? Let's try to see this thing. So what are virosomes exactly? Okay, so when we talk about these virosomes, guys, virosomes are going to be some lipid molecules. Okay, so you have lipid molecules called as liposomes. So you have liposome, and plus to that, you are going to have the inactivated virus. Okay, so now let me just tell you how this process really works, like what really
So,
happens out. Yeah, go on, go on.
I'm so sorry, sir. I had to unmute because I don't think so you're reading
I
the
can't hear you.
messages.
We can't hear you. We can't hear you.
Sir, are you reading the? I'm so sorry, sir. Are you reading the messages?
No, no, I'm not getting your messages only. Actually, I don't get the notification right now. I've updated the this thing, Zoom app, so I'm not getting it. Huh? Okay, okay. 1 minute, I'm checking your messages. Okay, but there's no problem. What is the question? I am not getting your question.
Actually, sir, the screen is going real blur in
Uh-huh.
between, and it's barely getting fixed, but not totally fixed. So it's become really difficult to read and write.
Guys, everyone else is facing the same problem. Is the screen blur or is it okay? Or is it the problem with her? Okay, others are saying it is fine. Himanshu is saying it is fine. Sia is saying it is fine. Just try to log out and log in again. Okay, just log out and log in again. Okay, yeah. Others are saying it is fine. There are many messages out over here who are saying it is just fine. Okay. The thing is, I have updated this Zoom app now. So now what happens is, like, if anybody messages now, I don't get a notification, or maybe I have not like explored the options out over here. So that is a little hassle out over here at this moment. Okay, so guys, let's try to see what is this virosome and how are we going to do this. So let's imagine we have a virus. Okay, so let's imagine this is virus. Now viruses come in different shapes, sizes. Okay, so let's imagine we are talking about this virus. Okay, now inside this virus, guys, we are going to have our DNA. Now it can have its DNA, it can have its RNA, it can be anything. Now, what we do is, see, we know for sure that these viruses can infect your cells, right? And they infect the cell, and then they're going to deliver this nucleic acid into the cell. So we are going to exploit this property only. So what we do is, this, whatever its DNA is, this is going to be removed out. Okay. So in the next case, guys, how are you going to see that? Oh, this is going to be completely empty kind of thing, right? If this is removed out, it is going to be completely empty like this. Okay, and now what are you going to do? You are going to take the vaccine. Okay, we had said either you take the vaccine or you take the DNA. So what are we going to do is, let's say this is either the vaccine or the DNA, whatever you wish to insert it into the virusome. Okay, so this is the DNA, or you can take vaccine. Now you are going to take this and you're going to insert it into this virusome. Immediately, what are we going to see? Okay, in the next case, this virus is going to have the DNA of our interest. And now, whenever this thing is going to like go and infect any cell, immediately the DNA of our interest is now going to enter into the cell. That way. Everybody getting this thing, guys? So this is how it is going to work. So if I have to show you the next stages, like, let's say we have our cell, but I'll draw the cell in different color. Okay, so let's say this is our human cell. Okay, so let's say this is our human cell. Now what happens, guys? This thing, it is now going to come, let's say, Okay, it starts infecting out over here. Okay, the moment it starts infecting, now you are going to see in the next case it is going to like deliver either the virus can enter or it can deliver this thing inside. Okay, either of the two. And then your DNA is going to come to the inside. Okay, and then now once it has come onto the inside, guys, now it is going to be used and it is going to merge with your the existing DNA, and then this cell can produce your own product. So let me just show, this is your own nucleus like this, and this starts producing its own product. So this way, so guys, this way we can introduce these virosomes in our body. Okay, this is first thing. Okay, second thing is like very simple. Okay, so just have a look, guys, if you're okay. Okay, very simple concept. You take a liposome, which is nothing but a lipid molecule, and you add an inactivated virus to it, then remove the DNA out and put the DNA of your interest inside of it, and then infect the cell with it. That's it. Okay, the other thing, guys, the second way we can introduce the genes is very simple. This we have always seen right from the start of the chapter. Okay, so the second way is going to be in vitro or ex vivo. Okay, so in vitro or ex vivo. So in this case, guys, what are we going to do is we are going to take our patient. Okay, and from this patient, we are going to remove the defective cells. Okay, either you remove the defective cells or you remove some normal cells. Okay, however it is. Okay, defective or normal cells, whatever you want. Okay, and now what are we going to do? Let's say we have taken those cells and we have put it into your test tube. Okay, so once again, this test tube is going to have all our cells. So let's say his cell is something like this. Okay, and he has his own DNA. Now, what are we going to do, guys? We are going to make use of our plasmid. Okay, so let's say this was the plasmid, and we have already constructed a plasmid with the gene of interest. Okay, we have taken our gene, we have put it into the plasmid, we have kept this plasmid ready. And now what are we going to do, guys? We take this plasmid and we are going to put it inside. The moment it goes inside, guys, now the plasmid is going to be out over here. Now what are we going to do, guys? We take these cells and now we are going to put it back into the patient. You put it back into the patient, guys. This is how it is going to work. And if this happens, then what, guys? Immediately you're going to see that because the plasmid is there, it starts producing the product of your interest. And plus, whenever this cell is going to multiply, once again, then a number of plasmids are also going to multiply that way. So this is why we're going to use the gene of interest. And this is the second method, in vitro or ex vivo. Okay, and the third one, guys, we have in vivo. Okay, now how are we going to do, guys? In vivo meaning what? Inside the organism itself, directly in the organism. So in this case, guys, what are we going to do? We take the gene of interest and we are going to directly put it into the cells of the patient. Okay, directly. Now, generally, we don't do this thing. This is a little tedious job, but directly put into the patients, guys. This is what is done. And how are we going to do this thing? We are going to use like, micro injections kind of thing, very, very fine needle force. And we will try to put it into the cell nucleus itself. A little tedious job, but then still, like, we can try to do this thing. Chances of this, it is questionable because you never know whether the cell will readily accept it or no, but still this can be done, guys. So these are the three different methods by which we can put the gene into your cell. Okay, so guys, just have a look. If everybody is okay, then fine. Okay, if anybody has doubt, then we can move ahead. Anybody has any doubts, guys? Anybody wishes to ask anything from here, this part? Okay. Okay, chalo. So guys, let's try to move ahead. Let's try to see the next thing, guys. Okay, now the next thing we're going to talk about is, see, when you introduce these foreign genes into the person or into the organism, what are these organisms called? Guys, these organisms, we call them as genetically modified organisms. Okay, or we will call them as GMOs. Okay, so genetically modified organisms, or we are going to call these as GMOs. Okay, but now guys, let's try to see how are these GMOs going to be and why are we going to use these GMOs? Let's try to see. Okay, so in one simple sentence, these are nothing but organisms whose genes have been altered. Whose genes have been altered and modified. And guys, why are we going to go for this? What is the purpose behind this? Guys, because maybe we want to introduce insect resistance into the plant. Okay, so for insect resistance. Okay, fine. Apart from that, guys, we can have drought resistance. Okay, so that the plants can assess, they can survive through the drought regions properly. We can increase nitrogen fixation, guys, by using this. Okay, apart from this, we can increase photosynthesis. Okay, then we can increase salt tolerance. Okay, plus we can increase your vitamins and nutrient content. Okay, along with this, guys, we can also increase their proteins and carbohydrate content. So for all these purposes, etc., 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 guys, we are going to make use of these genetically modified organisms. Okay, but now why will we go for these things? Like, why should we go for genetically modified organisms, and what is the purpose? So the first purpose is, guys, see, actually it is not an ethical thing. Okay, but then we have our own problems, and that is why in order to tackle those problems, we are going to make use of some of these. So first thing, guys, we are going to make use of transgenic plants. Okay, so what are these transgenic plants, guys? Transgenic plants are, maybe you have, like, introduced some kind of your gene. So for example, increase in photosynthesis, increase in protein content, okay, into the plant, and that is the resultant plant is going to be known as transgenic plant. So why should we go for this? See, guys, what happens is, see, in countries where there is high population and where there is a low amounts of agriculture, there is going to be tremendous stress on the food production. So how does food actually gets produced, or what is this? So if you look at food production will always follow arithmetic progression. Arithmetic progression means what? 1, 2, 3, 4, 5, 6, 7, 8, dot this way. So it will go in a stepwise manner. But guys, if the food is moving in such a slow rate, okay, is the population also going to move in this rate? No, the population moves in your exponential manner. Okay, or we say it is geometric progression. So if we talk about population will be 1, 2. Until here, it is going to be fine. But then 1, 2, 4, 8, 16, 32, 64, 128. So, guys, if you see, let's see, until 2, it is fine. But afterwards, you are going to see that there is going to be tremendous stress on the food. the food supply, because the more and more population increases, the less and less food is going to be available per capita that way. And in order to tackle this problem, guys, that is why we went on for your transgenic plants whose production is higher, whose rate of growing is high, whose properties are going to be much better than the normal plants. And by doing this thing, guys, what type of plants have we created? We have created wheat, we have created rice, we've created potatoes, okay, your maize, that is corn, etc., etc., etc. But now, is this really ethical? I told you, like, see, this is not really ethical and it should not generally happen, but we have our own problems. So guys, understand what happens. So let's imagine we have our original plant. Okay, so let's say you have created genetically modified maize. Okay, and let's say this is your maize plant. Okay, let's imagine. Okay, now this maize plant, it has its original DNA. Now please understand, okay, what I'm trying to say. This is our original plant, it has its own DNA. And guys, we have this genetically modified plant who has the new DNA. Okay, and this new DNA, guys, we have made this thing. Nature has not made this thing. So now what will happen, guys, when pollination is going to take place, are the bees or the the insects, are they really going to see, Array, this is original plant, this is modified plant, I'm not going to carry his pollen grains? No, it is not possible that way. So maybe what may happen is during pollination, the pollen grains may mix. So from the new DNA, it can go to the original DNA. And guys, what will this result into? If this kind of a thing happens, then this results into a mixture of organisms, mixture of your DNA. So guys, are we really going to look for, we don't even know, like, whether this DNA is stable. We don't even know that. But now you are mixing the organisms plus mixing of DNA. What does this lead to? This leads to contamination of gene pool. So if by any chance later on, let's say 10 years later, we identify that, oh, this thing is a problem, the DNA that we had introduced, it is causing a little bit of trouble, right? Maybe it is, like, inciting cancer in patients. Then can we still continue to use this plant? No, then we'll say that, अरे नहीं नहीं नहीं, इसका product नहीं खाने का, इसका product नहीं खाने का, इसका product नहीं खाने का. But then by that time, what will have happened, guys? Pollination and fertilization would have happened so many times that this DNA would have been incorporated into the new generation plants, and it will be difficult to retrieve the original DNA. And then you are stuck with it. You are stuck with it, and now you will have to keep on using it, or else you'll have to stop using the plant altogether itself. Are you getting the problem, guys? This is the biggest problem with your genetically modified organisms. Okay, so there was this company who was trying to do the same kind of thing, and this failed really, really badly. Okay, so there was this company called as Monsanto. So what this Monsanto was doing was, it was creating seeds in such a way that these these kids, the seeds basically, they would give you huge amounts of growth, but then they would not be able to reproduce. What happened initially, these people, the farmers and all, they were all crazy for the Monsanto seeds, and they were getting huge amounts of benefits also. But slowly what happened was, people who were eating their products, they started suffering from various kinds of diseases. Eventually it was found that the problem was with the Monsanto seeds. Immediately this company was shut down, guys, and then you saw that this company, it went bankrupt, shut down, and there were many cases which were filed against this, for doing some unethical work. So that way, so this is what happens, guys. This is how it works. But anyways, by that time, guys, we had already lost the gene pool, and if we continue to do the same, then we will lose all the plants. So that, so first thing is your transgenic plant. Okay, the second thing that we're going to see, guys, we are going to see your insect resistance or pest resistant plant. So this is also another important thing. Let's try to see. So we have insect resistance or pest resistance? Okay, so now let's try to see what happens in this case, guys. So when we talk about insect resistance and pest resistance, guys, this is one of the most important property, because you will always see that, whenever you have palms and all, they're always infested with pest or insects. You'll always have a problem with them. So there should be some way. So generally, what do these people do? They are going to make use of these pesticides, okay, or insecticides. These also have their own problem. Why? Because 50% of the pesticides or insecticides, they are just going to fly away. They are not going to reach the target. Apart from that, they are going to seep into the underground water, and then you're going to contaminate the underground water. Plus, these pesticides, they are going to kill all the germs, okay, all the pests, all the insects, invariably or indeterminately. Meaning they are not going to determine who is going to be like a good pest, who's going to be a bad pest. They are going to kill everybody. So there are many problems associated with your normal pesticides, like your synthetic pesticides. So we go on for another method, that is we will incorporate the insect resistance gene into the plant itself. So we don't have to make use of your insecticides or pesticides. And for this, guys, what are we going to go for? We are going to go for the most famous Bacillus thuringiensis. Okay, so we have this Bacillus thuringiensis, and guys, this Bacillus thuringiensis, it produces a toxin. This toxin, it is called as the Bt toxin. And which gene is responsible for this toxin production? This gene is called as Bt gene. Now, what does this Bt gene produce? Now, Bt gene, it actually produces your cry protein. Okay, this is the protein, crystal protein that we actually call it as, but this is known as cry protein. Now, guys, this Bt gene, it actually produces this cry protein, but this cry protein, it is actually inactive. At the moment when it is produced, it is inactive toxin. Okay, but now what happens, guys, see, whenever any pest eats this thing, okay, when this is ingested, okay, when this is ingested, now what happens is that inactive toxin, it enters into the gut of the organism. Okay, but into the gut, guys, what is the pH of the gut? PH of the gut is going to be alkaline. So now in the alkaline pH of gut, guys, this inactive toxin, this is immediately converted into your active toxin. Inactive toxin immediately converted into active toxin. And the moment we have this active toxin, guys, now this is going to cause a lot of problem for the pest. So immediately this is going to give rise to intestinal discomfort. Okay, so it is going to give you immense amount of pain. Okay, it is going to result into your cramps. Okay, this is going to result into huge amount of your contractions. Along with that, guys, it is also going to result into your loose motion kind of thing, like in the indigestion and all. And ultimately there is going to be tremendous amounts of swelling in the intestines, and now immediately the organism is going to die. So this is going to result into the death of the pest. So this is how we are going to target this Bt gene basically. Okay. But now see, this Bt gene was present in Bacillus thuringiensis. If we want to create a plant out of it, we need to remove this Bt gene and we need to put it. 1 minute, someone is on mute. Okay. So we need to put it back into your plant cell. So let's try to see how we are going to do this thing. So in short, okay, once again, we are going to use your plasmids and all only, but I'm just giving you right now how do we generally do this thing. So in normal cases, what are we going to do, guys? We know that, okay, this Bt gene is important. So we are going to remove, okay, we remove the Bt gene. Okay, if you want to be more specific, you can also call the cry gene. Okay, so you remove the Bt gene and now you are going to introduce it into the plants. Okay, just like our normal way. So, like how we are going to make use of your Agrobacterium tumefaciens, right? You take this cry gene or the Bt gene, you put it in the Ti plasmid in between your tDNA, and then you put it back into Agrobacterium. You take this Agrobacterium, you infect your plant cells. So, introduced into the plants. Okay, once this has been done, you allow the plant to grow. Okay, now the moment the plants grow, guys, each cell has the Bt gene. Okay, and because it has the Bt gene, it is producing toxin, each and every cell. Therefore, each cell produces toxin, the Bt toxin. Okay, now in this situation, now as the plant is growing, if for any reason, any bug, okay, if for any reason, if there is any bug, if there is any caterpillar, or if there is any moth, okay, anybody, okay, anybody who is a pest, when they are going to consume, okay, when they consume the leaves or other part, immediately you are going to see that this inactive toxin that is there gets converted into your active toxin. And once again, where is this going to happen? In the gut region. Okay, so in the gut and in the alkaline pH. And immediately you are going to see all those things. Once again, intestinal discomfort, swelling, and then pains, cramps. Okay, immediately this active toxin, it is going to be activated, and then this is going to result into the death of the pest. That's it. This is how it is going to work, guys. Okay, but now by using this method, guys, what have we created? We have created, first of all, BT cotton. This is the most famous one, guys, BT cotton. Second, we have BT brinjal. Third, we have BT ladyfinger bhindi. Okay, so ladyfinger. We have got BT beans as well, guys. BT beans, etc., etc., etc. So this is how we are going to make use of your BT gene, guys. Okay, so this is how we produce your insect resistant or pest resistant plants. Okay, just have a look, guys. If you're okay, then we'll move ahead with the next part. Any doubts, guys? Anybody wishes to ask any doubts from here? Anybody? Due to the toxin, gut becomes alkaline? No. Alkaline is the pH inside the gut. So you will see that in the stomach it is acidic, and in the intestines it is going to be alkaline. Okay, chalo, guys, let's try to move ahead. Okay, next thing. Okay. Okay, now the next thing, guys, what are we going to see? We are going to see the same thing. Okay, on the same lines, we have alpha amylase inhibitor gene. So alpha amylase inhibitor gene. Okay, so alpha A1PV. Okay, why do we call this as A1PV? I'll just let you know. Okay, alpha amylase inhibitor. So A1 actually stands for inhibitor. PV is because it is obtained from an organism, which I will just mention. So this is going to be obtained from Phaseolus. Okay, Phaseolus vulgaris. Guys, very, very important. Phaseolus vulgaris. That is why PV. Okay, and now what are we going to do, guys? Okay, so we are going to remove this gene. So here, the gene is removed from Phaseolus vulgaris. And now this is going to be introduced into tobacco plant. Okay, the moment it is introduced, and once again, how are we going to introduce? We take the gene, we put it into your TI plasmid, in TI plasmid, in between your tDNA, then that is going to be taken, put it into agrobacterium. You take the agrobacterium, you infect the tobacco plant, and then inside the tobacco plant, it is going to start multiplying. Okay, so once again, the moment it has this thing, guys, what are you going to see? Immediately, this gives the tobacco plant pest resistance. You are going to get pest resistance. And pest resistance against whom? We have two important bugs. So we have Zabrotus and we have the second one, that is your Cosobrucus. अरे रे, क्या हो गया? अरे रे, sorry, just a moment. Okay, Brucus. Okay, so guys, this is what is actually going to happen. Let me just quickly show you how do these look like. So see, this is one pest, okay, the Zabrotus, and this is your Calsobrucus. Okay, so something like this. So we get pest resistance for your tobacco from these pests. Okay, so this is one thing, guys. Okay, next thing, guys, let's try to move on to the next part. Next part is something that is called as biofortification. Okay, let's try to understand. What do I mean by biofortification? Okay. So guys, generally, you must have heard that, we have fortified salt, fortified stuff, fortified chips, or fortified, food products. So there is a little difference between what is biofortification and what is your fortification. Now, guys, first thing, the first basic difference is whenever we talk about fortification is whenever you have got the product. Let's say you have made the chips. Okay, everything has been done, like, it has been produced, it has been, like, cooked, it has been, like, properly fried and all, everything has been done. Then after that, what you do is you add a little bit of your vitamins, or you add a little bit of your some good compounds onto it, like, good products. That is called as fortification, when you are adding nutrients, minerals after the production of the product. That is fortification. But biofortification is when the plant itself is growing, you are going to give it so much amount of nutrients that it becomes really, really healthy by itself. That is biofortification. Okay, so guys, in biofortification, what is a normal this thing, normal aim? Our normal aim is to increase the nutritional value. To increase the nutritional value or to increase the nutritional contents of the plant. Okay, and by this, guys, what have we done? See, by doing this biofortification process, guys, we have treated night blindness. Okay, so night blindness. What is night blindness? This is generally caused by deficiency of vitamin A, guys. Okay, but we have tried to overcome this thing by having biofortified plants, which are rich in your vitamin A or provitamin A, that is your beta carotene. So that way. Second thing, we have also done this thing with rice as well. So we have taken the rice and we have made it like, it is increased or high in carbohydrates. Okay, we have converted it into such a manner that it is one which is high in carbohydrates and which is going to have a little bit of other things as well. Okay, so generally, normal tendency of your rice is what? First, okay, we have rice. Rice is rich in carbohydrates, that is there. But now, is it going to have any other minerals or any other nutrients? No. So it lacks other nutrients. Okay. But now what we do is to overcome this thing, guys, we have produced transgenic rice. Okay, so we have produced transgenic rice. But now, guys, what is the importance of transgenic rice? What we have done is we have introduced this vitamin A gene inside of it. And the moment you introduce this vitamin A gene, guys, from this we can get two things. Now, either you take this vitamin A gene, you add it into mustard, guys, you are going to get golden mustard. Okay, golden mustard, because this is going to be rich in your vitamin A. Same way, guys, what have we also done? We have also taken this and we have put it in your rice. So when you put it in rice, what do we get? We get golden rice. Okay, and what is the idea of golden rice, guys? This rice, it has increased the production of vitamin A. And why did we do this? We did this so that we could tackle your night blindness, guys. Okay, so this is how we tackled night blindness. Okay, everybody all right with this thing? So this is first thing, okay, in first thing in your biofortification. Okay, let's try to move on to another thing. See, the next thing, guys, it is going to be to increase your oil content. Okay, to increase oil content. So what are we going to do, guys? You are going to take the yes, Gatorade is a fortified thing. Yes. Okay, all your this thing, all your most of the fast food products and all, guys, those are all fortified. Okay, in fact, if you look at your salt, normal salt, we said, oh, no, normal salt contains iodine, table salt contains iodine. So how is that iodine added? It is added at the time of processing. So once again, that is also fortified salt. So if you look at the labeling packaging, you will always see that it is fortified salt. Okay, so guys, now what are we going to do is, see, we are going to take, okay, to increase the oil content, we take the oil producing gene from Arabidopsis thaliana. Okay, so this is this plant Arabidopsis, like Arabidopsis thaliana. Now you take this thing, and now what are you going to do? You are going to transfer it into the plants who are not producing good amounts of your oil. So we are going to put it into your soya bean. We are going to put it into palm. We are going to put it into your rapeseed mustard. Okay, we're going to put it in your sunflower. And immediately, guys, what are we going to see? We are going to see that because of this, immediately the oil production capabilities of these plant increases tremendously. So this is another thing. Okay, then next, let's try to see the third thing out over here. So the third thing is to increase iron content. To increase iron content of the plant. So now in this case, guys, what are we going to do? We are going to take, okay, we take the ferritin protein gene. Ferritin is nothing but your iron. Okay, so you are going to take the ferritin protein gene. And now, from where are we going to take this thing? Guys, we are going to take this thing from your soybean. You take it from soybean and the phaseolus. Okay, we have talked about phaseolus vulgaris, so same guy. Okay, you're going to take it from phaseolus. And now what are you going to do? Guys, to increase the iron content, we take it from this and we introduce into rice. Okay, the moment you have introduced into the rice, what are you going to see? This rice is now going to show increased iron content. And whenever you consume this rice, immediately that iron is going to be transferred into your body. So this is the next thing. Okay. Okay, chalo. But now let's try to see, guys. Apart from this, what else are we really going to do with your plants, guys? Okay, so when we talk about your plants, guys, we are going to do various things, okay, to achieve various results. So what we can also get is, in the same way, we can get herbicide resistant plants or herbicide tolerant plants, or we can also create disease resistant plants. Okay, another important thing is, guys, in the case of maize, what do we do so that the maize should not be attacked by pests? Okay, we have a very different strategy. We are going to create this maize who has low production of lysine, tryptophan, and methionine. Okay, and the moment we have this thing, this is not going to attract the insects. That is the plan. And because we have this thing and we're going to keep the other things rich in maize, by doing this, guys, the maize is going to be protected and the pests are not going to be attracted at all. Okay, another important thing that we can do is increase nitrogen fixation. How we can do this? By making use of your nitrogen fixation genes. So this can be obtained by transferring NIF, NIF. Okay, NIF genes is nitrogen fixation genes. Okay, and finally, one last thing that we can do is we can also produce stress resistant plant. Okay, stress resistant plants. These are the things that we can obtain by creating your transgenic plants, guys. Okay, everybody all right with this thing? Sure. Any issues, guys? Anybody wishes to ask anything from here? Just have a look, guys. From biofortification, just have a look. Okay, chalo. So guys, let's try to see. So guys, so this is like, how we were trying to get the plant itself modified, okay, so that it can, produce good things and all. So this is like, your transgenic plant. Okay, but now we can also do a little bit more thing out over here, guys. And what is that? We can modify the post harvesting techniques or characteristics. Okay, so let's try to see what is that. Chalo. So the next thing is modifying post harvest techniques or characteristics. Okay, so now guys, what happens is, see, whenever we talk about fruits, okay, fruits have a very small shelf life. Okay, especially like, you keep them outside and all, then immediately within like, 3 4 days, okay, they are going to go bad. And this is generally seen with the case of your tomatoes. You cannot keep tomatoes outside for a long time, neither you can store them for a long time. So there is an inherent problem with it. So let's try to see how is it going to happen. Yes. Okay, so guys, first of all, we are going to use this thing for fruits. Okay, uh, especially like, the fruits, they spoil during transport also, and during your normal this thing. So spoil, just a second, they spoil during transport or during your normal storage. Guys, my question to you is, have you ever, seen tomato which has gone bad, like a really really bad kind of thing? Okay, it smells awful. It smells like, your poop just, increase the smell to like, 10 15 times more. And it is that bad, guys. It feels as if, pura naak ka bal jal gaya. It's that way, guys. It is a really really horrible horrible smell, but then it happens. Okay, so guys, I want to save this tomato. I don't want it to go bad. So how can we do it? Okay, so first let's try to see like, points. Okay, so guys, when we talk about these tomato, okay, what is the inherent property that is like, spoiling it? So let's try to see this thing. So guys, tomato, it has a very low shelf life. And the reason for this is because it has an enzyme which is called as polygalactourinase. Okay, very, very important this thing. Okay, very, very important term. Polygalactourinase. Okay, now guys, what happens is, or we'll say this is PGU. In your exams, especially in your board exams, this is a very, very favorite fill in the blank, guys. Always ask. Now, what does this polygalactourinase do? This polygalactourinase, it breaks down the cell wall. So, as your tomato grows older and older, as it ripens, the breakdown of cell wall is initiated by PGU. Okay, so now in order to overcome this, now what have the scientists done? Scientists have like, introduced, okay, to overcome this, we have seen scientists, and scientists have introduced your antisense gene for PGU. Okay, scientists have introduced antisense gene for PGU. Okay, now what is this antisense gene for PGU do? So normal gene you have removed, and instead of that you have put this thing. Immediately, guys, this produces less amounts of your PGU. Okay, less amounts of P. Now, if by any chance it produces less amounts of PGU, guys, then are you going to see the breakdown of cell wall at the same rate? No. Then you are going to see that the cell wall is broken at a very, very slow rate. And in turn, what are we going to see, guys? We are going to see increase in shelf life because we have introduced this antisense gene for PGU. And guys, this entire plant that is going to be like, prepared by doing this, what is this called? This is known as flavor saver tomato. Okay, guys, remember the spelling. This is also very important. Okay, flavor saver tomatoes. This way. This is what we need to know, guys. Okay, about your plants. Okay, now the next thing we are going to use your plants is making use of them as factories for biopharmaceuticals. Okay, but what we will do is, guys, we'll just break over here for 5 minutes. Okay, it is reaching 8:15 right now in my watch. 5 minutes, 8:20, and once again we'll start with the next part. Okay, chalo guys, let's try to move ahead, guys. Okay, so guys, here we were seeing like how we are going to make use of your plants and how we are going to increase their contents. Okay, different content and how we are going to make it sustainable for a larger shelf life. Okay, but now the next thing that we are going to talk about is plants as factories. Okay, so plants as factories for what? Let's try to see. Okay, so we are using plants as factories, and this is for your biopharmaceuticals. Okay, but now how is it going to be? Like, what are we going to do? Guys, the very simple. See, when we talk about them being used as factories, we are going to produce what? So we are going to see that we are going to produce, okay, used to produce high amounts. High amounts of your carbohydrates. So carbohydrates, fats, proteins. Okay, along with that, guys, we are also going to get certain essential oils. Some of them, see, for example, rose has essential oil, tulsi has essential oil. So we are going to use them for giving us these essential oils as well. And generally, guys, what are these oils used for? Oils are going to be used for making your perfumes. Okay, for making perfumes. Okay, along with this we can also make use of like various chemicals which are obtained from the plants. Along with that, some kind of lubricants as well as some adhesives, etc, etc, etc. All this can be obtained from your plants, guys. Okay, another important aspect is the use of bioethanol these days. Now, what is this bioethanol, guys? Bioethanol is nothing but you are going to prepare ethanol which can be mixed with your petrol or your diesel, and it can be burned to generate energy. That is nothing but your bioethanol. And guys, this bioethanol, today this is a very booming field. We say this is your renewable source of energy. Okay, once again, we are going to use this thing. We're going to make this thing from your plant. And what is this? If everything works properly, guys, this has the possibility to replace fossil fuels. Just a moment. It has the possibility to replace fossil fuels, guys. Okay. Now, same way as we have got your bioethanol, guys, there is huge amounts of research which is going on biodegradable plastics as well. Biodegradable plastics. So all this thing can be obtained from your plants. Okay, so this is one thing. But now the second thing is, if we are making use of them as pharmaceuticals, then what? So let's try to say, in the case of biopharmaceuticals, okay, so guys, what are we going to produce by using them? We are going to produce certain proteins. We are going to make use of antibodies. We'll use them to prepare antibodies for us. Okay, to produce vaccines, drugs, etc. Okay, so all these things can be done. Now, this is one thing, but guys, apart from this, we can also produce certain hormones. So, for example, we have been trying to produce this growth hormone in tobacco plant. Growth hormone in tobacco plant. So also, guys, what have we done? See, we said antibodies. Antibodies, we have been trying to produce this thing. Okay, in what plants? in plants against your HIV. Okay, we have also tried to produce this against RSV, respiratory syncytial virus, or we have the herpes simplex virus. Okay, so we have done this thing, guys. And the last thing, okay, the last thing, guys, we have also tried to produce anti-cancer proteins as well. Okay, this is another thing that we need to know. Okay, but now, are these the only things that we have tried, our hands on plants? No, guys, we have tried many more things. For example, guys, we have tried making super glue in plants. So, for example, in which plant did we produce this super glue? This was produced in your tobacco. And guys, now what how did we come across this thing? Guys, this super glue was actually found in mussels. So what happens is in mussels, it attack mussels meaning your, those shellfishes like kind of thing. Okay, it attaches muscles to rocks. Okay, it attaches muscles to rocks. And what we found out was that, oh, this super glue can be produced in your tobacco plant. And once we get this super glue, guys, we can use this thing for body repairs. So that is super glue. Okay, another thing, guys, we have started our hands at producing oils, different types of oils. So for example, guys, what we have done is we have taken genes from marine algae. Okay, so we have taken genes from marine algae, which is nothing but a plant. And what have we done is we have tried to introduce this in your plants. And what do we get, guys? We get increased oil production. And oil like what, guys? Oil like your cod liver oil. So if you have, guys, ever taken 7 C's cod liver oil, 7 C's, or, you have those, fatty acids, omega-3 fatty acid tablets and all. Guys, this oil, cod liver oil, it tastes awful. It smells awful, guys. Okay, but then it is really, really nutritious. That is the other thing. Okay, so we can make use of oil from here. Guys, another thing, we can also produce drugs in the plant. Okay, so drugs like what? And this is very, very important, huh? Guys. So we can make use of drugs like artemisinin. Okay, artemisinin. Now, what is this drug, guys? This is like your anti-malaria drug. Okay, then, uh, okay, along with this, guys, if we talk about your drugs, we can also produce like, highly active. Okay, another one is highly active opium painkiller. So even this can be produced. Highly active opium painkiller. Okay. So this is next thing. Guys, apart from this, what is the next thing that we can do with your plants? We can create edible vaccines. Guys, you just chew onto the plant, and while chewing, all the components are going to be absorbed into your body. Once they are absorbed into the body, now what? You have got resistance against diseases. So guys, what have we created out over here? We have created potatoes. We have created tomatoes. We have created cereals out of this. And guys, we've even tried our hands on banana. Okay, so these are all the different things that we have done with regards to your transgenic plants. Okay, now guys, these were only plant things that we were talking. Now we have to move ahead onto your animals. Okay, so just have a look, guys. If anybody has any doubts from over here, then quickly ask, or else we'll move ahead with your plants. we'll move ahead with your animals. Sir, but after cooking, are you there in this thing? I'm trying to message you, but I was trying to answer your question, but it says that you're not in the meeting. Okay, your first answer to your first question, that yes, it can have an effect on the taste as well, because you are altering the DNA. So definitely it can have an effect on the taste. Okay, second thing, but after cooking, the vaccine in the potato goes away. I have heard so. Is it true? So actually, that depends upon how the vaccine is and what genes have you produced. So if you have uh, one minute. So if you have this thing, like if you have a gene which is heat resistant or heat tolerant, then definitely your vaccine is going to be saved. So once again, it depends. Okay, another thing is overdose of opium becomes an addiction. Yes, absolutely. And that is why you don't get it like normally over the counter kind of thing. Okay, because it is highly addictive. So yes, that is true. In fact, people get addicted to painkillers, also normal painkillers as well. So even that is a possibility. Okay, painkillers, sleeping pills. Okay, apart from that, you have anxiety, anti-anxiety medicines and all. People get used to it because whenever you take these medicines, you feel good. You feel ki, now you're relaxed. You're relaxed. In fact, the simplest example is Otrivin. Guys, if you have ever tried Otrivin is like really, really dangerous stuff. Okay, it is highly, highly addictive. So what happens is, like you take Otrivin and suddenly you will feel that your entire nose has completely opened up. Guys, you will feel as if you have never breathed so beautifully ever in your life. When first time I had used Otrivin, guys, I was like, oh my god, I don't know what is breathing. That was the kind of reaction that I had, because my nose was like completely clear, and I could see that oh my god, I can take good amounts of air. Earlier I was thinking that I am having some defect, but now, oh my god, okay, it is a wonderful feeling. But then what is the side now, what is the side effect? Guys, slowly, slowly you start becoming addicted to it, and afterwards it is going to start inflammation of your nose, and then what happens, it's going to result into pain. Now once again, what is going to happen, you are going to be completely addicted to it. Second thing, there is going to be inflammation. So, and eventually you will notice that anybody who's taking Otrivin, eventually you will notice that the small dose won't do any good to you. You have to keep on increasing the doses. So first, if you take one spray, if that was sufficient, afterwards you have to take 2 or 3 sprays, and that is always incremental, guys. So anyways, with regards to your opium and all, it's also is the same thing. Yes. Okay, so guys, the next thing that we are going to start off with, so whatever we talked about right now was with regards to your plants only. But just like, yes, 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 it is, yes, you have to take it cautiously, but then we don't follow all that stuff. Now, once you are addicted to it, you're addicted to it. So you don't take caution. You only tell me if you have been taking Otrivin, how many times have you been taking it like, as per the rules? No, you won't, right? Whenever you feel you you spray. So that way, it doesn't happen that way. But okay, chalo, guys, let's try to move on and let's try to see what are your transgenic animals. Okay, once again, the same thing, guys. Transgenic animals are nothing but these are the animals whose genes have been modified. Okay, so let's see what happens. Okay, so once again, if you want, I'll just mention this thing once again. So here, the entire genome, this is modified. Okay, but now, guys, why do we do this thing mostly? Why are we going to produce transgenic animals? Are we going to produce it to eat or to produce something? Yes, sometimes we do it, but most of the time, guys, this is generally made for your medical research. This is done for your medical research. So guys, what happens, in your like research, we want only certain genes to be expressed. So what are we going to see? You are going to have specific factors, and these specific factors, these are going to be used for certain gene expressions. So for that, we are going to use your transgenic animals. Okay. Apart from that, we are going to study your disease progression. Guys, let's see, for example, your polio. We have been studying polio in mice for a long time, guys. So we are going to use it for disease progression and disease detection as well. So disease detection, disease progression. Okay. Apart from this, guys, we also see study toxicology, meaning like if for any reason, guys, your whatever product you're having, it is toxic, when these compounds are given to these transgenic animals, immediately they will start showing a reaction, and they will show you that this product is toxic for life. That way, so it detects, these animals, they detect toxic agents. So guys, you know what, you, the males though, I'm sure you may not be, but females must be using all the cosmetic products and all, Lakme, L'Oreal, right? So you have that, something which you apply on your face and all, right? It changes your color. ऐसा वो क्या brown powder रहता है, brown powder. Then you apply something on your eyes. You give that something, someone, yes, foundation, right? Foundation, contour. Then वो आंखों के ऊपर you apply those, क्या वो, there is that color, हाँ mascara, okay, mascara, okay, eyeshadow, guys. So all that stuff, all that cosmetics that you use, guys, do you directly take that and apply it onto your mouth, like onto your face, like without even knowing that whether it is safe? No, guys, we need to first make sure that all these compounds are safe for your skin. Then only we can, like, market it into the, the country. Otherwise, what will happen tomorrow? You take that product, you apply it onto your cheeks, and suddenly you see there is, like, adverse reaction, pimples, and then there is, like, scaling of the skin. Would you really like it? No, we wouldn't like it. So all these products, guys, these are tested on mice, transgenic mice. The transgenic mice that we used in our labs were albino vistar rats. Okay, so that albino vistar rats, they're specifically modified in such a way that they will show increased reactivity to toxic products. So what we used to do, we used to, clear off their patches on the skin. Okay, so thoda sa patch, that hair is going to be removed, the fur is removed, and we used to apply those products onto those patches. Now, if by any chance, guys, if by any chance these products contained carcinogens, what used to happen? Immediately, where we were applying those products, that part would start showing tumors. What did it mean? That this product contains a carcinogen. Would you want to use such kind of a product onto your face? No. So, guys, all that testing, where has to be done? It has to be done on these animals only. So, for that, we use this thing, guys. Okay. Apart from this, guys, like I said, we are going to make use of them in pharmaceutical industry. This is the biggest thing. We are going to use this thing in the pharmaceutical industry, but for what, guys? We are going to use it for detection of your proteins. Either you detect proteins or you make proteins. Okay, we can use it that way. We can use organisms as it's like a reservoir for your proteins. See, for example, proteins or antibodies. I will just explain this thing. Okay, for proteins or antibodies. So guys, for example, if we want to produce your anti-venin, okay, anti-venin, or let's imagine we want to produce your antitoxin. Okay, antitoxin against clostridium. Chalo, let's say clostridium. So guys, you must have taken anti-tetanus injections, right? See, what do you do whenever you have, like, a cut or something? You go to the doctor, and what do you tell the doctor? Give me tetanus, TT injection. Guys, do you think that doctor is like crazy that you say you give me tetanus and he's going to take tetanus and he's going to put it into your body? No, 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 guys. He is actually going to give you anti-tetanus, right? So, guys, how is that anti-tetanus produced? How are those anti-tetanus antibodies produced basically? So what we do is, guys, we are going to take your horse. Okay, so let's say we have this horse. Okay, so let's imagine this is your horse. So you are going to take the tetanus germs, okay, and you put it into the body. The moment the tetanus germs, the Clostridium tetani enters into the body, now what? This horse is going to feel that, oh my God, he has been infected. Horse starts producing huge amounts of antitoxin against the tetanus toxin, guys. Okay, now what happens? Now, the moment the levels of antitoxin in his body increases, you are going to insert another dose of the germs. Immediately he feels that, oh my God, there is second round of infection out over here. He starts increasing huge amounts of your antitoxin production, huge amounts. Now what do? We remove a little bit of blood from here, purify the blood, separate the antibodies, and then we are going to take it as your antitoxin, that I mean your anti-tetanus injection. Getting this? So guys, this is what we are going to. So we use them as your we use them as your fermenters for us that way. Okay. Anyway, so just like antibodies, we can produce your anti-venin. Okay, anti-venin is against your snake venom, and also we can use it for your product testing. Okay, so it can be any product, like I said. It can be any industrial product, any product out over there. Okay, guys, like I said, we're going to use this thing for various kinds of research purposes also. So we do this thing with cancer as well. So in research for cancer. Okay, apart from this, guys, this is not the only field that we're talking about, like product testing and all. We can also prepare huge amounts of other things like, sorry, we can increase production. Production of what? Production of hormone. Second, we can increase production of meat. We can increase production of milk as well, guys. In the case of your fishes, now we can do this with fishes as well. So if we do this with fishes, what are we going to do? We are going to increase tolerance to their environment. That means either it's cold or either it is like hot temperature, etc. So it is going to be like adjusting it to the external temperatures. Guys, next thing that we are going to do is we can use pigs and other organisms, okay, other animals, for xenografting. Xenografting means what, guys? You can create some skin, and on top of this you can remove it and then you can put it onto your body. Okay, xenograft means foreign grafting. Basically, xeno means foreign and graft means a part. So we can use this thing, guys. Okay, so this is like just a gist of like why we are going to use these organisms for. Okay, but now let's try to see the most important ones. So guys, the most important ones of these transgenic things is your transgenic mice. Okay, so let's try to see how are these transgenic mice used for. Like very very simple. Transgenic mice, then we have transgenic farm animals, then we have cattle for this thing, and then we have others also. So slowly we'll start talking about each one of them. Okay, but first of all, guys, just have a look if everybody is okay until this point, guys. Okay, so guys, let's try to see how are we going to use these organisms. Okay, so the first thing we'll talk about transgenic mice. Okay, so guys, most again 99% of the times, guys, we are going to use these transgenic mice only. The reason is very simple. They have a very short life cycle. Within 21 days, you can generate whatever you want, guys. Okay, so that way. So why do we generally use them for, guys? We use them mostly for cancer research. Okay, we use them for cancer research. And guys, why? To detect the genes, maybe to detect genes responsible and their expression. To detect the cancer genes, maybe, or the genes responsible for cancer. And we also study their expression. Okay. Next thing, guys, we are also going to study breast cancer. Okay, with the help of these, we study breast cancer. Okay, and for breast cancer, guys, what we do is we are going to see the MYC and the RAS genes basically. Okay, you're going to introduce maybe this MYC and this RAS genes into your mice. And now, once inserted into the mice are later analyzed. Okay, you're going to put it into this mice and later analyzed. Okay, this is going to give you an idea how the disease is going to progress and what really is happening when you are struck with breast cancer. Okay, chalo. Next one. Okay, so next thing we are also going to use transgenic farm animals. Okay, so now guys, transgenic farm animals, as the name suggests, guys, this is going to be used for increasing the food products basically. So what are we going to do? So you're going to see it can result in increased meat production. It can result in increased milk production. Okay, it can result in increased egg production, guys. Okay, so other products like we can increase wool. We can also increase disease resistant in different organisms. So that way, guys, so this is how we are going to make use of your transgenic farm animals. Okay, but now let's try to see especially cattle for food production. Okay, so let's try to see. So guys, for this food production, what are we going to generally do? Okay, we are going to look for bovine meaning your cattle. Okay, bovine, beta casein, and we have kappa casein. Okay, so beta casein and kappa casein. Okay, now what we do when we take these genes, okay, when we take these genes and we insert this into your cattle, guys, immediately you are going to see that the milk production increases tremendously. Okay, so milk production increases. But guys, what are we going to see? You are going to see 80 to 20, 8 to 10, 20 folds. Okay, you are going to see when we do this thing, 8 to 20 folds increase in beta casein in the milk. And if you look at your kappa casein, around 2 folds increase in your kappa casein. This is what is going to happen, guys. So that is why we are going to use cattle for your food production. Okay, then next thing, guys, we are going to use these animals for therapeutic production, human therapeutic production. So let's try to see what else are we going to do out over here. Okay, so for human therapeutic production, what are the different compounds that we are going to produce? Okay, so guys, the first one we are going to produce human lactoferrin. Okay, so these are the certain compounds that we are going to be producing, guys. Okay, so lactoferrin. Second thing, we have human alpha lactalbumin. Okay, so lactalbumin. Okay, next. After that, we have serum albumin. Okay, serum albumin. Okay, then we have lipase. Guys, when we have seen that, when we take these products and we put them in cows, you are going to see increased production of all these things, guys. Okay, another thing that we have been doing this, guys, is generation of factor 9 for your treatment of hemophilia. So what do we do in this case? Let's try to see. So guys, we have factor 9. Okay, what we do is we take the genes basically. Okay, we are going to introduce it into the cows. Okay, from the cows are going to have udders. Udders are nothing but basically the mammary glands. Okay, and now the mammary glands, the moment you see this thing, guys, what are you going to see? You are going to have increased production, increased production of that factor 9. The moment we have increased production of factor 9, now what, guys? Whenever you have the product, you are going to be treating your hemophilia. So this is going to be used in the treatment of hemophilia. Okay, same way, guys, what we had done was we had introduced some increased protein genes in your Rosie. Okay, Rosie was the first transgenic cow. Okay, and in the Rosie, what we saw was there was increased protein production in the milk. That is what happened with Rosies. Okay, another important thing, we also use this kind of method to produce antibodies. Okay, so these are some of the things that we do in your human. So human therapeutic production. Okay, chalo. So next thing, sorry, we are going to make use of transgenic sheep. Okay, okay. So what are these sheep going to be used for? So we are producing transgenic sheep so that we can get increased amounts of your wool production, increased wool, increased growth, increased meat, okay, etc. Along with this, guys, what we are going to do, how are we going to tackle this thing? That, how are we going to increase the production of your wool? So, guys, what we generally do is we are going to take bacterial genes. Okay, and bacterial genes what like your cis E. Okay, and the second one we have is cis M. So we have cis E and cis M. We take these genes, and what are these going to do? These are going to help us in production of your cysteine. Now, guys, the moment you have increased amounts of cysteine, immediately you are going to see that you are going to have increased amounts of keratin. And guys, where is keratin going to be found? It is going to be found in your hair. So here, in the case of your sheep, what is this going to result into? Immediately, this is going to result into increased wool production. That way. So that is transgenic sheep. Okay. Next thing, guys, out over here, we have transgenic pigs. Okay, I told you, see, transgenic pigs, they are going to be used for xenografting, meaning you can produce whatever products of human into your sheep, into your pigs, and then you can once again use it into humans. That is what is going to happen, right? So, just a moment. Okay, so let's try to see the last one out over here in this category, transgenic pigs. Okay, so first of all, guys, why are we going to go for pigs only? The reason for this is, guys, pigs are going to be almost the same as humans. If you look at their anatomy and humans' anatomy, they are really, really close in the arrangement of your organs, in the way the things are functioning in your body, almost the same as humans. And that is why we will go for this transgenic pigs. So these are going to be similar to humans, and that is why we'll go for this. Okay, but now why will we go for transgenic pigs? So there are few things. First, because I want increased meat products. So guys, for example, if you love bacon, okay, bacon comes from pigs, so you need good products out over here. So for that, this thing. But now I told you the second thing we're going to use it for xenografting. Okay, so like what we will do. So we will use them for developing organs. Okay, so for example, guys, if we look at your heart, and from the heart, if we look at the walls, guys, we can generate heart walls in your pigs. And these walls can then later be used to treat humans. This is also possible, guys. Okay, so this is transgenic pigs. Okay, then we have two more. We'll just quickly finish that part, guys. Okay, so nothing like different as such. Okay, we are going to have transgenic fish. Okay, so let's see what are these going to be used. So guys, transgenic fish like what? Salmon. We have trouts. We have catfish. Okay, we have tilapia. Okay, all these are like uh, like commonly eaten fishes. Okay, so what are we going to do, guys? We are going to introduce certain genes for your growth hormone. The moment we introduce these genes for growth hormone, guys, what are you going to see? Suddenly their size increases. And guys, more the size, more is the market value, right? So this is how it is going to be. Oh, just a moment, guys, just a moment. Okay, so guys, we are going to use this to increase the size. Okay, apart from this, guys, we are going to use like to increase the taste and all. Okay, we can also make use of your chicken crystalline protein. Chicken crystalline protein. Along with this, guys, we are going to make use of another genes which is going to introduce E. coli hygromycin resistant gene, meaning it is not going to be affected by E. coli. So that way, so E. coli hygromycin resistant genes. Okay. So this is, and then the last thing out over here, guys, we are going to have your transgenic chicken. Okay, so now why are we going to go for transgenic chicken? Because we are going to have increased meat production. Now, should we go for this? No. Okay, why? Because this is not something that is natural. So you're going to have increased egg production. You are also going to see increased resistance to diseases like coccidiosis. Okay, like the poultry diseases that we have talked about. Okay, so we have coccidiosis, we have viral diseases, all these things, guys. Okay, so we are going to be resistant to these. Okay, also, guys, okay, in your sorry, in your fish also we will have few more things. Okay, let me just put it down, I forgot. Okay, so we can also introduce other genes in your fish, like, to increase cold tolerance. Okay, we can also use to increase oil content, or we can also use genes to increase fat content. That is. So this is what we need to know with regards to all your transgenic animals. Okay, next time we have a very small part that is remaining, that is what is bioethics and bioethics, biopatents, and what is biopiracy. So that is very small. Okay, so we will do it in the next class. We'll finish off with the chapter, and guys, at the same time we will start with the MCQs. Okay, chalo. So guys, once again, we'll stop over here for the day, and I'll see you in the next class. Till then, take care. Bye bye, everybody. Bye bye.